Hello, everyone, and welcome to today's presentation titled Robotic Automation of Cell Isolation to Streamline Cell Therapy Manufacturing. This webinar is part of the second annual event in the Cell and Gene Therapy virtual event series. My name is Delia Osborne with LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of today's presentation. If you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's go ahead and get started. I'd like to introduce today's speaker, Dr. Fred Parietti, Director of Protein Sciences at GenScript. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Dr. Parietti, you may now begin your presentation.
Thank you very much. It's great to be here. I was looking forward to this webinar. My name is Fred. I'm the co-founder and CEO of Multiply Labs, and we develop robotic manufacturing systems for next generation therapies, and in particular for cell therapy manufacturing. In this presentation, I will be presenting and discussing our technology that we designed for enabling the manufacturing of gene-modified cell therapies at industrial scale. In particular, we believe that robotic technology and advanced robotic technology is really needed to enable the scaling of cell therapy manufacturing beyond the current throughput level that are achieved with manual processes. So a quick overview of our technology to begin. We designed our robotic technology to achieve 4 key features. First, we wanted it to be compatible with market-leading GMP instruments. In other words, we are not designing new instruments or instruments that are different from the market-leading instruments that cell therapy companies are already using. And by doing this, we do not force a cell therapy company to change their process. Instead, we teach our robots to use the instruments that they would be using in a case that they're already using. The second key feature is flexibility. The idea here is that our partners are free to choose and to keep using any combination of instruments that they're already using. The robots will adapt to that through a Lego-like modular structure, where you just need to select the modules containing the instruments you are already using. We also really prioritize being future-proof, meaning that we keep adding new instruments to our automated portfolio, so to speak, so that if a new instrument or a new technology becomes available in the future, we can add that to the robotic system. And finally, of course, the goal is to be cost efficient in order to decrease the cost of goods sold for cell therapies, but also in order to increase the throughput. So let's get more in detail about how all of this works in our robotic systems. So first of all, here's an overview of the market-leading instruments that we have automated and that we are working to automate with their respective manufacturers. And of course, particularly relevant to today's presentation is the GenScript CytoSinct 1000, which is the solution that we have automated for cell isolation and enrichment. And it was very, very important to automate market-leading instruments because, of course, it takes a lot of engineering effort to teach a robotic system, a robotic arm, to operate an instrument that's been designed to be used by human operators. And so it's very important we start from instruments that are recognized as industry leaders, so that the first nucleus, the first core of instruments, which you see on this slide, will be useful to automate as many processes as possible. And so as you can see, it's already end-to-end in terms of the capabilities of our systems for automating cell therapy manufacturing. And we've actually published data about the performance of our robotic systems in the use of these instruments. More specifically, we have shown that the robotic systems are equivalent to the manual use of these instruments. And you're going to see more detailed data in the course of this presentation. Now, it's very important to give you a visual point of reference about how do the robots actually perform all of the complicated tasks that are needed to carry out a cell therapy manufacturing process. And you can see it here. We're going to now play this video. Perform tasks that normally would be performed manually by a scientist or a manufacturing operator inside of a clean room. And so you can see here a kind of like a sampling of very different tasks, from vial to syringe transfer to open close incubator to the resuspension of bags or of a G-Rex bioreactor, all the way to handling cartridges that contain more advanced consumable kits. For example, the consumable kit for the CytoSinct 1000 by GenScript. So it's very important to underline that the robotic systems are performing these tasks autonomously. And so, for example, they stage an instrument by clicking in its cartridge, and then they remove the cartridge once the process has been completed. So both staging and line clearance are performed by the robotic systems. Now, going a little bit more in detail on how one of these tasks works and how do we automate the operation of very complicated, and very advanced instruments, I'm gonna move to slide number 7 here, which is actually about the CytoSinct. So as you can see on the left, the CytoSinct, the instrument that you all already know and use, is left unchanged. This is very important because we can't change the instrument or we would change the process. So instead of changing the instrument, we put the instrument inside what we call a robotic module. And a robotic module is, imagine a cube of about 1 meter or roughly 3 to 4 feet of side length. And inside this module, there is the original instrument. But in addition to the original instrument, there is also a set of electromechanical auxiliary systems. And this module makes it possible for a robotic arm to operate the original instrument without changing it in the same way that a human operator would use it. It's very important to underline that on the software side, we just talk to the original, unmodified instruments. And so we just need to be able to send commands and receive data from it. On the hardware side, however, what we do is repackaging the original manual consumable kit with the same tubes and the same bags. And so this kit is then repackaged into a rigid cartridge. And why we do this? Because robotic systems are not very good at handling soft, unstructured systems like tubes and bags. So we leave the tubes and bags unmodified. It's the original consumable kit. But what we do is we put it into a rigid box, right? So now it's the best of both worlds. The cells are still handled in the same exact consumable kit as before, right? So according to the cells, there's really no difference. But from the robotic system point of view, the robot is now not handling soft, unstructured systems. It's handling rigid cartridges and cassettes. And so can handle a rigid cassette in a very, very repeatable way, in a very, very precise and accurate way. We do this automation, we do this process of starting from an instrument and then creating a robotic module for all the instruments that you saw in the slide before, slide 4. Here it is, and essentially, moment, going back to slide, fantastic, and so you can expect basically a set of multiple modules, each of them containing at least one instrument, right? Now, we're going to go to slide number 6, because slide number 6 shows a typical configuration. So as you can see, a robotic system, and we call this system a robotic cluster. The robotic cluster contains a high number of of modules, typically about 15 to 20 independent modules. But which modules to use is actually selected by our partner, the cell therapy company. And so if a cell therapy company is using the CytoSinct, they're gonna use at least one CytoSinct module. If they need many CytoSinct, many CytoSinct instruments to be operating in parallel in order to increase throughput, well, no problem. They're gonna add multiple CytoSinct modules, right? And the same can be done for all other types of modules. Keep in mind that if we have developed a module that you don't need as a cell therapy company, you don't need to use it, right? That module can not be included into the final configuration of your robotic cluster. So we really designed this module-based system to be extremely flexible and extremely efficient. Okay, so now, before showing actually the next video, I wanted to briefly hop onto this slide, slide number 9, because these shows you a section view of the robotic cluster, right, of our robotic system. Here you can see that in the middle of the robotic system, there are multiple robotic arms. In this picture, you can see 2 robotic arms on a single rail on the floor. But in our most recent configurations, we actually have 4 robotic arms at the same time. 2 moving on a rail on the floor, and actually 2 more on the ceiling. And I'm pretty sure this is not the maximum number of robotic arms we can use in the system. It really depends on the bottleneck. In most typical processes, the bottleneck tends to be the incubator. And so we've made a lot of work to fit into a single incubator as many bioreactors as possible. In the figure here, you can see we can fit into a single incubator up to 18 separate 1-liter G-Rex bioreactors. But there is also another bottleneck for essentially most manufacturing processes. It's the number of robotic arms. And that's why we moved from a single robotic arm in our initial prototypes all the way to 4 robotic arms. in in our most recent version of the robotic systems, and we might as well go even higher than that. And so now that we know how the system works, I wanted to show you the video here on slide 8, which brings together everything we've discussed so far into a kind of vision of how Okay, here we are. So what that video showed is the robotic arm at the center of the robotic cluster moving cartridges containing reagents or cells or consumable kits from one module to the next. And so you could see in the video the robotic arm handling all of the typical reagent and consumable kits that are handled by manual operators during a standard cell therapy manufacturing process. So the robotic arm was handling syringes, vials, bags, a G-Rex bioreactor, and also more complicated consumable kits, like, for example, the consumable kit for the CytoSinct 1000. Essentially, what happens thanks to this technology is that the entire end-to-end cell therapy manufacturing process is automated inside of a cleanroom system, which is the robotic system itself, that does not contain any human operator. And so by doing this, we can achieve at least three big improvements. One, more sterility, because the biggest source of contamination in cell therapy manufacturing processes is actually human error, typically. So this is removed because now you have robots that are, can be clean and decontaminated, and they don't breathe, they don't generate particles. They do not touch, what they're not supposed to touch, and they have very repeatable trajectories. So it leads to higher quality and lower standard deviations. And that's what we've seen in our published data. The second benefit is a reduction in costs. Of course, the robotic systems are more efficient and can run a large number of processes in parallel. They can also ensure that every single instrument around the robotic system is saturated so that there is no bottleneck instrument. If there was a bottleneck, for example, a robotic arm or an incubator, we would add more copies of that bottleneck in order to solve it through parallelism. The third significant advantage of robotic systems is throughput. Specifically, we have seen throughput that can be higher, up to a couple of order of magnitudes more than a typical throughput in a manual suite. So the benefits of the use of robotic system are very, very significant. And we have a slide here to show them, to kind of visualize them, right? And so, a normal manual serial process has, all the manufacturing processes one after the other. However, the robotic systems can be highly parallel. And so we were mentioning before that we can fit up to 18 separate products, up to 18 separate lots into a single incubator. And we've seen cases with a very large number of incubators, right? So some partners have requested a very large number of incubators. And so if you have a lot of incubators and each incubator contains 18 individual lots, then the number of lots that you're able to manufacture in parallel really increases by orders of magnitude. And this is, a specific benefit of the robotic systems that can be so dense, so high throughput, but also they're still able to manage the really incredible complexity of processes where you have dozens of essentially lots being handled at the same time. If you were trying to do that manually, the risk of basically making a mistake, right, and confusing or switching two different consumable kits or vials would be very high. But it's not like that with the robots, because of course we design them and we simulate their behavior even before executing a process to ensure high reliability. The next slide kind of shows you the data. And the data that we are getting is really showing that the robotic process is statistically equivalent to the manual process. And here I would like to draw your attention to the left section of this slide, where we are showing the data that we have achieved by automating, together with the GenScript and Stanford LCGM team, the operation of the CytoSinct 1000. And so this is specifically referred to the cell isolation unit operation. And you can see in the graph that T cell purity post unit operation is actually not statistically different between the manual process and the robotic process. And this is from cells of the same donor. So it was very important to essentially run these set of experiments and publish the results to give our customers the confidence that the technology works as intended. And as of now, we are the only company that has published data about the performance of robotic systems for the automation of cell therapies. As you can see, we also have other examples, specifically cell counting, resuspension of bioreactors, and there's many more examples actually. And feel free to go in the paper to to see the details for yourself. This is a sort of recap, a high-level recap of the benefits of the robotic systems. In a traditional manufacturing suite, you're going to typically need a lot of operators, while with a robotic system that's automated, you only need one supervising operator, and the operator doesn't need to be in the same room as the robot. It could be remote. In terms of space, a normal suite tends to be pretty large, while a robotic system occupies much less space. And then in terms of scale, i.e. the throughput, well, you go from just a handful of products in parallel, managed manually, to much higher, level of throughput, typically even a couple of orders of magnitude higher, because of course, you can use multiple robotic incubators in parallel. And so in this case, 18 products per incubator, 54 products in parallel would simply mean that you're using 3 incubators, but you don't need to stop there. You can use more than that. These completes an overview of both our technology and our collaboration with GenScript about automating the CytoSinct 1000. And so I'm looking forward to the questions from the audience. Thank you very much for your attention.
Thank you so much, Dr. Parietti. We would like to thank you for your presentation today, and we will now open the floor for questions. If you have a question that you would like to ask, please do so now. You can submit that question using the Ask a Question box, which is located at the bottom of your screen. We will get through as many questions as we have time for. So let's go ahead and get started. Our first question is, personalized cell and gene therapies have historically been slow and prohibitively expensive to manufacture. What is the biggest bottleneck in manual production, and how exactly do the robotic clusters address it?
Thank you very much. This is a very good question. I would say the biggest bottleneck right now is automating a lot of manual tasks that are actually poorly documented. There are, I would say, there's a long tail of 10 or 20% of tasks in every process that we see. And typically we see very advanced processes. So there is a 10-20% of those tasks that is not really well documented, which is, these are tasks that are executed by very experienced scientists, but the scientist has kind of like learned almost implicitly how to carry out this task. And there's not really a precise record of the trajectory that should be followed, or a precise set of instructions or numbers that determines the timing for the process, right? It's really very manual, almost artisanal. And so can you teach to learn dozens or maybe hundreds of these tasks to a robot? This is really important to scale. And so that's why we're pioneering an imitation learning initiative, where we are actually using demonstration data, human demonstration data, to teach the robots how to self-learn and auto-learn, essentially, new tasks. So yes, that is the biggest challenge, and that's how we're tackling it.
Perfect. Thank you so much. The next question that we have received asks, in practice, how does this technology change the work of scientists and lab technicians? Does it free them to focus on higher value tasks or fundamentally reshape the skill sets needed in this field?
Yes, this is very important. Typically, scientists and technicians are very happy to see the robots in their facility, because the robots do the most boring tasks. The robots do the clean room tasks that are very repetitive and at the same time nerve-wracking, because they are high throughput, high repetition. And so usually skilled scientists and operators, they really enjoy to be involved in basically process development, to discover a new process and optimize it. But then once it's been discovered and optimized, it's really a robot job to do it over and over 24/7 at the highest speed and at the highest throughput. And so what we've seen typically is a pretty enthusiastic reception. Also, from the point of view of the operators and scientists that operate into a clean room, because nobody likes to be in a clean room, and everybody is pretty happy to basically be supervising the robots from outside.
Sure, makes sense. Absolutely. Thank you. And in taking a peek at the time, it looks like we have time for just one more question. Looking ahead, what do you see as the next frontier for robotics in advanced therapies? Will this model scale beyond blood cancers into other areas like rare diseases or regenerative medicine?
Yeah, definitely. We see a huge potential, mostly because pretty much, I would say the vast majority of next generation therapies, whether they are cell therapies or gene therapies or, every next generation bioprocess tends to be extremely complicated. And whenever we see that complexity, making that product for millions of people, right? Or at least, tens or hundreds of thousands of people, not just for one or two, we see that as a fundamentally a robotic task. And so that's really why we're very excited, about the future. Now, of course, we're seeing so much demand in the cell therapy space, and most approved cell therapies right now are in the oncology applications. But we're seeing so many more applications in regenerative medicine, autoimmune diseases, right? And even beyond cell therapy itself, And we think that robotics and automation have a role to play in all those areas.
Sure. Thank you so much. And again, we are at the end of our presentation. Unfortunately, I'd like to again thank you, Dr. Fred Parietti, for your time today, for your important research. Before we go, I would like to again thank our audience for joining us as well. I'd like to thank our sponsor, GenScript, for sponsoring today's webinar. Please do visit the GenScript booth in the exhibit hall after our webinar for more information and resources. Any questions submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand for 1 year until October 15th, 2026, and LabRoots will email you when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until then, take care, everyone. Bye-bye.